Hello. Hey, everybody. Who is with me today? Give me a hey in the chat if you're here. Let's see who we've got. So, obviously, this is another one of our drop in sessions. So, you can ask anything related to your A level A level or GCSE biology. I suppose this is mainly aimed at A level. As usual, we tend to have more year 12s, year 13s here than GCSE students. Hey, Kira. How are you? What have you been up to this week, Kira? Has this been your half-term holiday week? Or are you back in school? I can't remember. Anyone else here? It's just going to be me and Kira. Kira, you're going to get like one-to-one tuition. You're here, but you're not here. You've got to keep your streak of being here every week. Yeah, you've been doing your EPQ and biology. Oh, so you've been working even though it's been half term. Hi, Emma. Hello. So these sessions, Emma, are just free drop-in sessions for A-level biology. We can just chat about how you're getting on. We can answer your questions. We can go through specific topics. We do offer a full course. What year are you in, Emma? Are you in year 12 or year 13? I'm guessing you're A-level. So we do offer full courses. So if you're interested, you can get in touch with the team at My Edspace. Yeah, so we've got a year 12 course running at the moment. It started at the beginning of September, but you can join anytime. You get 2 lessons per week. You get a homework with each lesson. The lessons are recorded. You get monthly assessments, and it runs all the way through right up until the end of year 12. And then we've got a year 13 course as well. Hi, A, how are you? A few more people joining the chat. This is good. I had a few comments asking for a bit of help with respiration, but I'm aware that if you're in year 12, you haven't really done respiration yet. So we'll see. We'll see if anyone else has got any questions about any other topics. Because if you're in year 12, Umair and Kira, I know you're in year 12. A, remind me, what year are you in? Oh, don't worry, A. A, are you in year 12 as well? Or are you year 13? Year 13, yeah. So you might want to do some respiration. I don't know whether Umair and Kira want to do some respiration, get a little bit ahead, learn some year 13 stuff. But that's what I had planned. You're doing a 1 year intensive. Okay, so you're trying to cover everything in 1 year. That for sure is going to be pretty intense. So what have you covered so far, eh? What topics have you done? Kira's going to watch tomorrow when you get home. Perfect. Okay, well, what we'll do, mainly for A, but also for Amer and Kira, who are going to catch up. Kira's going to catch up tomorrow. We'll go through a little bit of aerobic respiration revision, I think, and then hopefully more people will watch this back and get to revise it with us. And you, Kira, have a nice Halloween. Off to a Halloween party. That sounds good. You're doing EdXL. You've finished the year 12 content. Bye, Kira. So you've already finished the year 12 content, even though you're just doing it in the space of a year. That's very impressive. So you might be moving on to respiration then pretty soon if you've not done it already. Let's go through some of the key details for respiration. I've just prepared a few slides. So there are 4 stages in aerobic respiration. A or Umair, I think Kira's gone, but do we know the names of the 4 stages? Obviously at GCSE, we just learn it as like one reaction, but in reality, it's a lot more complicated. So there are actually 4 stages that we break it down into. And we have to know the details of each individual stage. We have to know the name of the stage, and we have to know where it takes place in a cell. So, okay, so A's actually got one there, yeah. So the first stage is glycolysis. Perfect. The second stage is the link reaction. The third stage, which actually people find quite complicated, is the Krebs cycle. Yeah, you've got that one, eh? And the fourth stage is oxidative phosphorylation. There you go. You got 3 out of the 4 there. Oxidative phosphorylation, which does involve an electron transport chain. So that stage is kind of similar to photosynthesis, because the first stage of photosynthesis also uses an electron transport chain. So there are a lot of similarities there. If we go through and add in where the stages take place, so glycolysis actually occurs in the cytoplasm, not the mitochondria. It's in the cytoplasm of the cell. And you may also know that glycolysis also takes place with anaerobic respiration. So that stage does not require oxygen. So glycolysis occurs with oxygen or without oxygen in both types of respiration. The link reaction is then in the mitochondria, and it's in the matrix. The Krebs cycle is also in the mitochondria, same place in the matrix. And the electron transport chain also in the mitochondria, but this time it's on the cristae, which is the folded inner membrane. So if you think back to GCSE, you will remember that at GCSE you did say that aerobic respiration happens in the mitochondria. So all of these 3 stages are aerobic only. So they'll only take place if there is oxygen available. Whereas glycolysis, it's in the cytoplasm, it's kind of separate. So we see glycolysis with aerobic or anaerobic respiration, okay? So that's just the names and the locations of the stages. Is the matrix the fluid? Yeah, I've actually got that on the next slide, so I'll show you that now. So this is like an A-level diagram of a mitochondrion. We know at GCSE it's the site of aerobic respiration, but now we have to be able to label this organelle, think about its structure, and think about where the different stages take place. So if I label this for you, we've got the outer membrane of the organelle, but we've also got the inner membrane, which is folded, and the folds are called the cristae. Now, between those 2 membranes, you've got the intermembrane space. So that's all that is. It's just the space between the inner and the outer membrane. So one key thing to remember is that mitochondria do have a double membrane. Okay, A. And then this one here, A, would be the matrix. So the stuff that I've highlighted in yellow, which is like the fluid that fills the organelle, that's what's called the matrix. You've got one of the folds labelled here. So this would be the cristae. So that's just the term that we use for the folds of the inner membrane. Is there any process that occurs within the intermembrane space? Yeah, we're going to talk, well, actually, we're not going to talk about that tonight, but yes, when we get to the final stage of aerobic respiration, Tabor, it involves an electron transport chain, which is on the cristae, but it also involves hydrogen ions being pumped into the intermembrane space. So that's where you'd mention it, the final stage of aerobic respiration. The hydrogen ions, or protons, get pumped into the intermembrane space. So you get a really high concentration of hydrogen ions in the intermembrane space. So it is worth knowing where that is and being able to label it. The ATP synthase enzyme is on the cristae. The hydrogen ions get pumped into the intermembrane space, and then they diffuse back into the matrix through the ATP synthase enzyme. That's very complicated. That's the final stage of aerobic respiration, which may be the most difficult one. Do you guys know what this circular thing is here? Or what these little dotty things are here? Because this is a similarity that chloroplasts have. They have their own what? Same as chloroplasts. Yeah, hey Zaina. Yeah, so these are ribosomes. So mitochondria actually have their own ribosomes, and so do chloroplasts. And they are 70S. Well done. So they're smaller. Well done. So they're smaller than the ribosomes in the cytoplasm of the cell. They're like the prokaryotic ribosomes, yeah, 70S prokaryotic ribosomes. And then this is a loop of DNA. So it's very much like a prokaryotic cell in that it has its own DNA, circular DNA, and it has its own ribosomes. And they are 70S ribosomes, same as a bacterial cell or a prokaryotic cell. What do you think that enables the mitochondria to do? If they've got their own DNA, so they've got their own genetic material and they've got their own ribosomes, what does that enable the mitochondria to do? Or the chloroplast, because the chloroplast has that as well. Does anyone know? Good, exactly, Zaina. They can make their own proteins. Yeah, protein synthesis. So super clever organelles, if you like, they can actually make their own proteins. So for example, mitochondria are able to make their own enzymes for respiration. Chloroplasts can make their own enzymes for photosynthesis because they've got their own DNA and they've got their own ribosomes, which is where protein synthesis takes place. So in terms of the matrix, so there's two stages of respiration that happen in the matrix, and that's the link reaction and the Krebs cycle. So they're going on in the matrix, and then on the cristae, which is the folded inner membrane, that's where oxidative phosphorylation occurs. So it's important that we can state specifically where each stage of respiration takes place. So cool, all of that is in a place we can't, yeah, if you think about how small this is, I mean, we can't even see cells, and this is a small organelle in a cell. So yeah, absolutely tiny. I mean, you can just about see mitochondria with a really good light microscope, but to be honest, you kind of need an electron microscope to be able to see them because they're that small. But yeah, so that's our labeling. Now, I just wanted to show you, I can't obviously cover the whole of respiration. When I do this in my course, we would spend like at least 3 hours on the reactions, maybe even 4 hours going through all the reactions, all 4 of them. But I've just included detail of the first stage if you want to go through that with me. So this is what glycolysis looks like. Now remember, glycolysis is not in the mitochondria. It's in the cytoplasm, and it's the first stage of respiration. And it actually occurs in aerobic and anaerobic respiration. So this can occur even if oxygen is not present, doesn't need oxygen. So I've got some notes on the board. Now, I know you don't have the handouts, but I mean, you can write them down if you want to, if you think it's useful, or you can just kind of go through them with me. Because you could get like a 5 marker just on this first stage. So we do have to learn quite a lot of detail. So the first thing that happens is glucose is phosphorylated. Do we know what phosphorylated means, or can we work it out from the picture, from the diagram? You can see it happening here. This is where the glucose is being phosphorylated. What does that mean? Phosphorylated. Anyone have a guess? Yeah, good, Saina. Yeah, gets a phosphate, gains phosphate. Anytime you add a phosphate group to something, it's called phosphorylation. Exactly, A, exactly, Tabor. Now, glucose is actually phosphorylated twice. So we're actually adding 2 phosphate groups to the glucose. And you can see that it actually comes from ATP. That's where the phosphate comes from. So what we do is we hydrolyze ATP. And you can see it happening here. We hydrolyze ATP, or break it down. We break it down into ADP and an inorganic phosphate. And we add 1 inorganic phosphate to glucose, and then we add another inorganic phosphate to glucose. So it actually gains 2 phosphates from 2 molecules of ATP. And what that forms is a molecule called hexose bisphosphate. But you can, if you're doing AQA, you can just call that phosphorylated glucose. Phosphorylated glucose, which is a lot easier because the glucose has been phosphorylated, therefore it's phosphorylated glucose. Okay. Now, phosphorylated glucose is very unstable. So when you were in year 12, you might have actually learned that if you add phosphate to anything, it makes it more reactive or more unstable. And that's what we're doing here. We're adding 2 phosphates to glucose. It makes it so unstable that it immediately splits into 2 molecules of triose phosphate. So tell me, guys, how many carbons in a glucose? Let's start with that. How many carbons in a glucose? And we're going to split it into 2 triose phosphate. So then you should be able to tell me how many carbons in a triose phosphate. Hey, we got someone else in the chat. Welcome. Yeah, so glucose has 6 carbons. Obviously, the chemical formula is C6H12O6. Yeah, it's a monosaccharide. It's a hexose sugar. We know that from year 12. Once we phosphorylate it, so we add our 2 phosphate groups, it's so unstable that it splits into 2 triose phosphates. Now, each triose phosphate has 3 carbons. So we're not losing any carbons. We're literally just splitting the phosphorylated glucose in 2. So we've now got 2 triose phosphates. The triose phosphates are oxidized. Now, in this case, oxidation can mean different things, right? It can mean that you gain oxygen, or it can mean that you lose electrons or you lose hydrogen. But here, it's oxidized because it's losing hydrogen. So oxidation is loss, or oil rig. Oxidation is loss of hydrogen in this case. Hi, Aria. We're doing a little bit of year 13. I don't know if you want to stick around and learn some year 13. But yeah, it's losing hydrogen. You can actually see it here. It's losing hydrogen. Now, the hydrogen is accepted by NAD to form 2NADH. This is where it gets a little bit confusing, but the diagram's quite good. So those hydrogens that come from the triose phosphates are accepted by NAD, which is what we call a coenzyme. And that forms 2 reduced NAD. Now, 2 reduced NAD, we can call them NADH if you prefer. Don't worry, Aria. Thanks for jumping in, saying hi. So yeah, the triose phosphates lose the hydrogen, and the hydrogen is accepted by NAD. So we make NADH because it gains hydrogen. The end result is it produces 2 molecules of pyruvate, and this reaction does also make some ATP, only a small amount. So in fact, 4 ATP are produced, but remember, 2 ATP were required at the start to phosphorylate the glucose. So what we say for glycolysis is we get a net gain of 2 ATP. See you soon, Aria. See you in our year 12 lessons. Yeah, Aria is on the year 12 course with us, and Aria was here for GCSE as well. Aria is like a long-term student. Yeah, so we get a net gain of 2 ATP. Now, this seems super complicated, but I'm going to show you like a model mark scheme answer in a second, so we can simplify this down a little bit further. But basically, we're using 2 ATP, we hydrolyze them, we phosphorylate the glucose, we make 2 triose phosphates, we oxidize the triose phosphate so it loses hydrogen. Hydrogen is accepted by NAD, so we make NADH. The end result is 2 pyruvate, and we see a net gain of 2 ATP because we make 4 ATP, but we use 2 at the start, so we say there's a net gain of 2 ATP. Yeah, the courses are on YouTube, so it's similar to this, but with the courses, you get a workbook in advance of the lesson, so you have all of my notes and all of my diagrams, and we go through lots and lots of exam style questions. And with the main courses, you get a homework for every lesson, and then at the end of each month, you get a monthly assessment. and you get video solutions that I filmed to talk you through every homework and every monthly assessment. So yeah, it's a bit like the summer school, except the lessons are longer and there's more homework, assessments and things like that. Do we have lessons today? Let me think about what lessons I've got today. I've got Key Stage 2 and Year 13 today. Can't remember, Riju. You're Year 13, no? Are you Year 13 or Year 12? I lose track. Year 12. No Year 12 today, because what day is it? It's Thursday. So we're Monday and Wednesday, aren't we? Is there holiday lessons in Christmas? Not over Christmas, but there will be lots of revision sessions over Easter. And there'll also be cram courses like the night before the exams. But yeah, Christmas, we actually have a 2-week break. So there are no lessons at all over the Christmas holidays. But if you want to join us over Easter, there's going to be like 3 full days of biology A-level over Easter, like 3 intense days where we cover all of year 12 and 13. Perfect A, yeah. Basically what we do in those sessions is just go through like as many questions as possible and then use it as an opportunity to revise the topics as well if we struggle with any of the questions. We do essay writing skills, we do statistics, we do evaluation questions, we do practical questions, and just try and cover as many as we can from each different topic. So yeah, keep an eye out because we'll probably be advertising those quite soon and we'll have them for all the science subjects, maths, that kind of thing. Okay, so yeah, let's just summarize what we've learned. So for each molecule of glucose, what does glycolysis produce? Well, the end products, we end up with 2 molecules of pyruvate. We end up with 2 molecules of what we call NADH or reduced NAD. And we also get a net gain of 2 ATP. So glycolysis doesn't make a lot of ATP. And if you think back to GCSE, you do remember that because you should remember saying that anaerobic respiration releases less energy. That's what we say at GCSE. Well, it's because with anaerobic respiration, this is the only ATP we actually get. So it's very, very inefficient. You only get a net gain of 2 ATP with anaerobic. Whereas with aerobic, after glycolysis, you get the link reaction, you get the Krebs cycle, and you get oxidative phosphorylation. So you make loads more ATP if oxygen is available because you can then go through these other 3 stages as well. Let me show you an exam question, guys. So this was like a 5 marker. So you might just get a question on this very first stage of respiration. And this would be a mark scheme. So it's a little bit simpler. I explained it in slightly more detail just because I was trying to get you to understand it. But these would be the marking points. Yeah, I remember thinking that as well because you kind of just learn one equation, don't you, for respiration? You learn one equation for photosynthesis. And then when I got to A-level, I did feel like I was lied to. I suppose they just massively simplify it for GCSE, right? And then for A-level, you like hear the full story. Do you know what I felt like the biggest lie was, though, was chemistry. It's been a long time since I did it, but do you remember when you learn about electrons, like electron shells in chemistry? And what do you learn at GCSE? You say something like, well, there's 2 on the first shell and then there's 8 on the other shells. Is that right? It's been too long since I did it. But then at A-level, it's like, that's not true. And you've got all these other shells to think about and this like mad electron arrangement. Yeah, I can't remember the A-level version, but I remember feeling like I've been lied to. Definitely. So key marking points then for glycolysis. We've got phosphorylation of glucose. Obviously that requires ATP. It actually uses 2 ATP because we need 2 phosphate groups to phosphorylate the glucose. That makes triose phosphate, 2 triose phosphates. Then we oxidize the triose phosphate or remove hydrogen because oxidation is the loss of hydrogen here. That forms pyruvate. Again, it's actually 2 pyruvate if we're being picky. We get a net gain of ATP, or we could say we make 4, but we use 2. So it's a net gain of 2 ATP. and we make reduced NAD, or we can call that NADH, because when we say reduced NAD, we basically mean NAD has gained hydrogen, it's been reduced, or we formed NADH. So the mark scheme's a little bit simpler. So obviously you have to learn this kind of level of detail. Orbitals, see, I don't even remember what that is. But yeah, that's glycolysis. That all happens in the cytoplasm. So we're not even in the mitochondria yet. But after this, if we just kind of link it, pyruvate is actively transported. So that actually requires ATP, that requires energy itself, actively transported into the matrix of the mitochondria. And then we can have the other stages of aerobic respiration. But yeah, if there's no oxygen, it's just glycolysis. So we only get a net gain of 2 ATP. And then glycolysis just keeps getting repeated and repeated until there's enough oxygen available for it to go into the mitochondria. I'll just put the diagram back on the board here for you so you can see. We got any questions about this? It's difficult, isn't it? Because I know some of you are only year 12. So for some of you, this probably seems mad and like really hard. But trust me, by the time you get to year 13, hopefully you'll be so solid with all your foundations from year 12 that this will be fine. You'll be able to do it. That's why in year 12, we do all of our biological molecules, we do all of our cell stuff, so that in year 13, we can start to look at more complex processes. Because by this point, you know about ATP, you know about glucose. It should make sense. You know about oxidation and reduction, you know about the mitochondria. So it should be okay. Any other questions? I know we've just focused on respiration, but are there any other questions, any year 12 questions you want to go through quickly? And we can always go through the other stages for this as well if you think it could be useful, especially for UA. What do we think? Are we enjoying A-level biology? Are we finding it easy? Are we finding it hard? I'd be interested to know how it compares to your other subjects, actually. Like, is it one of your easier A-levels? Is it one of your harder A-levels? What did I think when I did it? I always thought it was one of the harder ones, but I did enjoy it. My teacher was really, really good. And I always found it really interesting to learn about like the human body and all like DNA and all that stuff. You're barely surviving. Oh no, Zaina, what are you finding hard? A says it's one of your easier ones. What other subjects are you doing then, A? Are you doing things like maths and physics? Because I'm guessing I would find those really hard. Hi, Pranav. Zaina, why are you struggling? What topics are you struggling with? Yeah, and I suppose biology is quite good for that. Like there's a lot of information, but if you're quite a motivated person and you put the time in, then you can definitely learn it. Maths, chem and bio, yeah. And I suppose maths and chemistry, it's a bit more applied, a bit more mathsy, so maybe that's a little bit harder. A lot of content compared to the other subjects. Yeah, we hear that all the time, way more content. And then the exam questions can be quite difficult, can't they? Because you think you've learned the content, you think you're good, and then the exam question has it in a totally different context, so it can be really difficult. Zaina, it's fun, but for some reason I always do bad memory. Oh, yeah, that is tough. There's so much to remember in biology, isn't there, Zaina? Just make sure, like, what I would do is after each lesson, like say in school or college, after each lesson, just read through your notes again and check that you understand it, and then make a really condensed set of notes on that lesson. And don't forget to ask your teachers if you don't understand anything. Don't kind of plod along, get to the end, and then panic. Try and do like little sets of revision notes regularly and revise regularly. You're thinking of choosing physics for A-level. I didn't do physics, so I can't really comment on that. I think if you're good at maths, though, you're going to be good at physics, no? Physics seems to be just like applied maths, so a lot of people do maths and physics, don't they? Yeah, summarize notes. Like sometimes it's hard to revise from your school notes because you've got loads and loads of stuff that you've done in the lesson. But if you take your school notes and you condense it into like the key information, and then by the end of the year, you'll have a little set of condensed notes on each topic. And obviously, we need to do past paper questions as well. Physics is a lot of maths. Yeah, so that wouldn't be for me. I did biology, chemistry, psychology, and sociology. So I had kind of two quite challenging sciencey subjects and then two subjects that were a little bit more written, I suppose, more essay based and a bit more where you could debate stuff. But I did find the biology and the chemistry harder because there was more I had to learn. But yeah, I never did physics. Last time I did physics was GCSE. I used to study and learn from the textbooks, couldn't absorb the info. Yeah, textbooks can have too much in sometimes, can't they? Chem, physics and maths and bio. Yeah, so you're doing like the four hardest ones there, I'd say. ARAM's doing the hardest subjects. What's the first topic in A-level biology? It depends. Schools teach them in different orders, but it's probably going to be biological molecules or cells. Or some schools have two teachers teaching in parallel. So you might be doing biological molecules at the same time as doing cells. Some schools just start with cells because they think it's easier because it kind of builds on GCSE cell structure. Personally, I prefer biological molecules because I like the biochemistry. Yeah, Zaina, yeah. Year 12 is so important. And sometimes when you're in year 12, you don't really appreciate that. Um, but by year 13, you kind of have to remember everything from year 12, don't you? Because it suddenly ramps up to complex processes like this. And you've got to kind of remember all your molecules and all your cell stuff from year 12. Is biological molecules like enzymes? That's one. So we do carbohydrates, we do lipids, we do proteins. So when we do proteins, yes, we do enzymes, but we also do ATP, DNA, RNA, water. We just cover kind of all the key molecules in living things and we look at their structure and function. So I really like it. Yeah, content is so heavy. Can you give a breakdown on the practicals we need to know? So there's basically 12 required practicals, but then there's also extra practicals that come up a lot in the exam as well. So I wouldn't just learn the required practicals. I kind of can't go through all of them right now, but required practicals include things like the root tip squash in mitosis. There's the osmosis prac with potatoes. There's the cell membrane prac with beetroots. There's an enzyme prac with like temperature or pH. There's a dissection practical. There's a microbiology practical where you have to grow microorganisms and test like antibiotics and stuff. There's a photosynthesis chromatography practical, yeah. But then sometimes there'll be other random practicals on the exam. So I would look at other random practicals that have come up in the past as well. And I try to cover them in my lessons. Like say when we do respiration, we always look at the respirometer. Now that's not a required practical, but it's come up on the exam quite a lot. So I'll try and include all those extra practical skills in the lessons. Pranav, I think for space related jobs, I'm guessing physics and maths maybe. What did I study at uni? I did biology. I just did straight biology because if you did biology, you could pick from all the modules. So I could just pick the ones that I found most interesting. So I mainly picked anything that was human anatomy related or anything that was like genetics related. I did a really cool module on stem cells. I did a really cool module on evolution and inheritance. My favorite though was one about developmental biology where we looked at how the embryo developed and like what determined its body plan, what determined its symmetry, what determined when different organs formed. It was all about the development of the embryo. That was really interesting. But then randomly, I also did one about the ancient Egyptians and the process of mummification, just because I read the description and was like, this sounds so interesting. It was like how they preserved the bodies, how they stopped the decay process. So it was kind of history linked with biology. how do you go about learning the practicals? So I would look at the required practical methods and focus on things like make sure I know what the independent variable is, what the dependent variable is, what the important controlled variables are. But yeah, it's hard. The best way is to just look at past paper questions, I'm going to be honest, and see what kind of things they ask you. Quite often it's just like drawing a graph from a table of data, or even drawing a results table, making sure you have the headings with the units correct. But they might ask you why you carry out certain steps, like why did you put the cotton wool in the test tube? And it's to prevent evaporation. But you see the same questions coming up quite regularly. So if you've looked at a lot of past paper questions, which you can easily get online, then you should be quite well prepared for them. Okay. Nice little chat. Nice little chat, guys. What's a good way to revise for A-level biology? Well, currently, I'm obviously going to sell myself, but currently what we're actually doing, I don't know if you guys have noticed, but on TikTok, we are going through the spec in order. So we post like 1 or 2 videos every day, and we are going through it for year 12 in order, because obviously that's going to help year 12. But if you're in year 13, you can also go back and revise year 12 with me. And then over here on YouTube, we're doing some longer videos, but again, we're going in order. So you should be able to get videos kind of to help you go through year 12 or revise year 12. Save My Exams is good for study notes. I mean, I always rate just like a CGP revision guide, just for concise notes. You know, for the test for proteins, why do we need to hydrolyze? That's not the test for proteins, Anna. You're talking about the test for non-reducing sugars like sucrose. Protein test is dead simple. Protein test, you literally just add bioret reagent and it goes purple. But Anna, you're talking about the non-reducing sugar test. So you have to boil it in acid to hydrolyze the glycosidic bond in the sucrose. And then you neutralize it. Then add your Benedict's in heat. And then, because you've hydrolyzed the glycosidic bond, you've now got what's sucrose made from? You've now got glucose and fructose, right? They are now reducing sugars, so now you'll get a positive result. Okay, right, guys, I'm gonna have to go. I've actually got some lessons to teach. I've got a Key Stage 2 lesson. Bet you all wish you were back in Key Stage 2. We're doing food chains, nice and simple. None of that in A-level biology. But yeah, thank you for a lovely little chat. I mean, we did a little bit of work, but then we just had a nice chat, which is always good. So yeah, lucky chaps. Lucky. They don't know how easy they've got it. Yeah, keep an eye out, because I'm not sure what time we're going to be doing these sessions in November. We've actually thought about maybe switching them to a lunchtime and doing like a lunchtime study club. So you can kind of access it on your lunch break in school or college with your friends. But yeah, if you keep an eye on my social media, we'll obviously post like a day or 2 in advance so you can see what day and what time it's going to be. But seeing as I have a lot of lessons in the evenings, we're thinking maybe we'll pick like 1 lunchtime every week and always do it at the same time. And then you can join from school. If you're allowed your phones, that is. Okay, right. Yes, Pranav, see you on Monday. Have a lovely evening. Have a lovely Halloween, whatever you're doing. And then it's November. Can't believe it's November already. Thank you, Zaina. Bye. Thanks for joining me. Bye, guys.